then, in this TOXBANK tutorial, we'll look at some of the key elements of good cell culture practice, which is fundamental to delivering good in vitro cell-based toxicology assays. During this presentation, I'll look at the introduction to the importance of good cell culture practice, potential impact of failure to adopt this kind of guidance, supplementary guidance that is available from other publications and related publications, and also some of the related TOXBANK information resources. The publication which I'll refer to for these, the first key principles for good cell culture practice, came from Kirker et al., and you can see the full list of authors in this slide, which identified 6 principles of good cell culture practice for use in any research or testing or production laboratory using in vitro cell culture or tissues. The first principle in good cell culture practice is probably the most fundamental and important one, and that refers to the need for an understanding of the in vitro system that you're working with and the factors which can affect it. Starting off with considerations relating to the cells that you're going to use in the assay, the authenticity of the cells, that is, are they mixed up with other cell types or switched, and identity of the cells, and also the provenance, meaning the traceability to the origin of the correct cell culture. And this can include in your own laboratory the confirmation of genotype and phenotype of the cells to make sure that you're using the correct kind of cell. Secondly, and also importantly, is the stability and functional integrity of the system. This can be affected by suboptimal cell culture passaging, and is stability and retaining the functional integrity of the cells is supported by setting up master and working cell stocks, which help you to minimize the number of passages that you subject cells to and enable a reproducible supply of the same cells over quite long periods of time. Finally, in this section, it's also important to recognize that new cell culture environments and culture conditions can have significant effects on the cell phenotype and their response to compounds. This may include changes in the media composition or growth factors, levels of serum supplements, gas levels in the cell culture environment, and obviously the temperature of the cell culture environment. Finally, also another key issue is to ensure, or try to ensure, freedom from microbiological contamination. In particular, mycoplasma contamination, which is very common in research cell lines. Also, environmental contamination with fungi. And there may also be issues with persistent viruses which can sustain themselves within a cell culture without necessarily showing a cytopathic effect. And these may also impact on the functionality of the cells. And of course, there may also be spores which have survived from other media or conditions, which fungi or bacteria, which may then proliferate at a later stage. Principles 2 to 6 cover a range of other issues. Firstly, quality assurance, controlling the cells, reagents and equipment that are in use to make sure they're consistent and the correct type. Documentation, to ensure that you can trace and replicate work that's carried out and that others can do the same. Fourth, it relates to safety on the laboratory and the environment in which you're working. And there is an appendix in the GCCP document on the use of Class 2 biological safety cabinets. And then fifthly, laws and regulations relating to handling of genetically manipulated organisms, chemicals, radioactive materials. And there are also laws and regulations on the use of human tissues and embryos, and the CITES International Agreement on use of cells and tissues from endangered species. These laws and regulations given in the 2005 publication may have been updated since then, so it's worthwhile checking your local conditions from a regulatory and legal perspective. And finally, there's also a section on education and training. It's very important to have a formal program for all personnel in the key procedures involved in cell culture to promote high quality work and safe operation. The appendices, as I've already mentioned, includes one on use of biological safety cabinets and also a section on safe handling of liquid nitrogen and its storage facilities. There are also some other relevant tutorials and publications available through TOXBANK. Glyn Might gives a presentation on access to TOXBANK, these TOXBANK reports in the Biomaterials Wiki. Glyn Stacey gives a presentation on mycoplasma contamination. There is also a deliverable report on points to consider in the acquisition of human cells and tissues under TOXBANK deliverable 4.6, and this can be located by following the early part of Glyn Might's tutorial as indicated above. Finally, there's also general information on the UK Stem Cell website at the indicated web address. There are a number of other useful publications you may wish to refer to. The one that I've already mentioned, the Kirker et al. Publication on guidance on good cell culture practice. There's also one specifically on banking and testing of human pluripotent stem cell lines published in 2009 in Stem Cell Reviews and Reports. And there is one on general good practice in a cell culture laboratory and setting up a stem cell laboratory by Inamidar et al. From 2005. And finally, a very recent publication has updated guidelines for the use of cell lines in biomedical research, which was published in the British Journal of Cancer in 2014.